For today's discussion, our topic will be the tests for carbohydrates. Let us first define what carbohydrates are. Carbohydrates are widely distributed in plants and animals, and they have important structural and metabolic roles. So for us humans and for animals too, carbohydrates are our main source of energy. And majority of the carbohydrates that we need or that we consume are being produced by those photosynthetic organisms, and majority of which are plants. Carbohydrates, in a chemistry standpoint, can be described as polyhydroxyaldehydes and ketones. So if we will be looking at the structure of carbohydrates, we will be seeing a lot of OH groups, and then the other functional group can either be an aldehyde or a ketone. Glucose is the most important carbohydrate in a human standpoint, and they are the major metabolic fuel of mammals. They are also, or glucose is also the precursor for synthesis of all other carbohydrates in the body. Classification of carbohydrates. So we have three classification of carbohydrates, which can either be a monosaccharide, oligosaccharide, or a polysaccharide. When we say monosaccharides, these are those carbohydrates that cannot be hydrolyzed or that cannot be further broken down into simpler carbohydrates. So the examples are trioses, tetroses, pentoses, hexoses, or heptoses, which are all aldoses. Now, when we say monosaccharides, these are actually the building blocks of the carbohydrates that we see or the macromolecules that we are seeing. Specific examples would be glucose and fructose. Now, the next classification is what we call oligosaccharides, to which the disaccharides belong to. So, oligosaccharides are actually formed by 2 or 10 monosaccharide units that are connected together by what we call a glycosidic bond. Now, the third classification of carbohydrates are what we call as polysaccharides. Now, these are the very large carbohydrates, and they are condensation products of more than 10 monosaccharide units. So, our specific examples would be starches and cellulose, which is the structural framework of plants. Now let us move on to the qualitative tests. If you can still remember from the previous experiment that we did, we have observed the results of the color reaction tests or the qualitative tests for carbohydrates. Now, if you will be looking at this is an example of a flow chart or a schematic diagram that will guide us to determine an unknown carbohydrate. Now in our experiment, we have observed the different results for the different qualitative tests of carbohydrates for our 4 sample carbohydrates, which are glucose, fructose, lactose, and sucrose. Now, in our first part of the carbohydrate experiment, we know what our sample carbohydrates are. And each of these sample carbohydrates have a specific result for each specific test. Now, for example, we are given an unknown solution or an unknown carbohydrate. Or for example, we do not know yet if our carbohydrate is, or our sample is a carbohydrate or not. Now, the first test that we should be doing is Molish test. Now, if we can still remember, Molish test is a general test for carbohydrates. So that will help us to find out whether our sample solution is really a carbohydrate or not. Now, if you will be looking at this figure or this schematic diagram, so if we will be doing the Molish test, there will only be 2 types of results. It will either be no reaction or a purple product will be formed. Now, upon doing the Molish test, if there will be no reaction, no color changes, or no ring that will be produced, then that means that our unknown solution is not a carbohydrate. Now, if there will be a purple product or a formation of a purple ring in between 2 layers, then that means that our solution or that our unknown is really a carbohydrate. So Molish test will confirm that our solution is a carbohydrate. Now, this one or the iodine test, this is is a specific test that will help us to figure out what type of carbohydrate do we have. Is that a simple carbohydrate or a complex carbohydrate? Now, since our four samples, the four choices of carbohydrates that we have are lactose, glucose, fructose, and sucrose, then it will be expected that they will not give any reaction or no color change, no formation of a blue-black complex will be observed if these four sugar solutions or carbohydrate samples are present in our unknown. But if what we have is a starch, then our result will be the formation of a blue-black complex, and that will confirm that a starch is present. Now, for example, our unknown is any of these four, then there will be no reaction. So that means that what we have is a simple carbohydrate. It can either be a monosaccharide or a disaccharide. Now, after that, what is the next test that we can use? So the next test that we will be using is the Barfis test. So in Barfis test, upon testing for Barfis test, if within the 2 to 3 minute range, there will be a red precipitate that will be formed, then that means that what we have is a monosaccharide or a reducing monosaccharide. Now, if, for example, there will be no red precipitation either after the 3 minutes or after 10 minutes, then that might mean that what we have is a disaccharide. But if there is a precipitation after 3 minutes, usually it's within 5 to 8 minutes or within the 10-minute range, but after or beyond the 3 minutes, then that means that what we have is a reducing disaccharide, and so on. So that is how we are going to be using this schematic diagram. So if you will notice in here, so for example, within the 2 to 3 minutes, a red precipitation occurs, then that means that what we have is a reducing monosaccharide. Then that can either be glucose or fructose. Now we can proceed with the Seluwanovs test because that will serve, or Seluwanovs test is a confirmatory test for ketohexoses, specifically for fructose. Now here, for the Seluwanovs test, if a blue-green product to pitch product, then that means what we have is an aldose, or if there is no change, then that means it doesn't have a ketose. Now, if a cherry red product is produced, then that means that our unknown sugar is a fructose. Okay, so now let us discuss into details the specific qualitative tests for carbohydrates. Let us first discuss Molish test. So Molish test, as I have said a while ago, is a general test for carbohydrates. So for example, if you want to figure out your unknown solution and you have an idea that it might be a carbohydrate, to confirm that, then what you should be doing is a Molish test. Now, the principle behind the Molish test is that when carbohydrates are treated with concentrated acids, specifically concentrated sulfuric acid, carbohydrates undergo dehydration to give furfural derivatives. And because of this, compounds condense with alpha naphthol to form a colored product. And that colored product that will be produced is the purple ring formation in between two layers of liquids. So here, we can see the formation of a purple ring. So this one is our purple ring. So an appearance of reddish violet or purple colored ring at the junction of two liquids is observed as a positive result for the Molish test. So all carbohydrates will of course give a positive result for Molish test. Our next test is the iodine reaction, or the iodine test. Now, this one is a test for complex carbohydrates or polysaccharides. So, in the laboratory, we did this for starch. And for your DIY home experiment, you also did this in the food samples that you tested. So iodine forms a coordinate complex between the helically coiled polysaccharide chain and iodine centrally located within the helix due to adsorption. So because of this, the color obtained will actually depend upon the length of the unbranched or linear chain available for complex formation. So the color that will be produced will actually determine or will actually depend on the type of complex carbohydrate that is present. So these are the positive color that will be observed when these complex carbohydrates are present. So for example, if amylose is present, which is a component of starch, then it will give a deep blue color. Now when amylopectin, which is also a component of starch, it will now give a purple color. Now, since amylose and amylopectin are both components of starch, then the color that we will be observing is somewhat a blue-black or close to purple or very dark purple color that will be observed. Now, for glycogen, so this is our storage form of carbohydrate that we can find in the liver. So if glycogen is present, then it will give a reddish brown color. Now, for dextrins, like for example, we have amylodextrin, erythrodextrin, acrodextrin. So for amylodextrin, it will give a violet color. For erythrodextrin, it will give a red color. Now, for acrodextrin, there will be no change in color. So these are the different colors that will be observed depending on the type of complex carbohydrate that is present in the solution. Our next test is Barfis test. Aldoses and ketoses, which are our carbohydrates, so specifically our monosaccharides, they have the capacity to reduce copper 2 ions even in acidic solutions. So actually, Barfis test is used or can be used to distinguish reducing monosaccharides from disaccharides. So for example, if you are not sure whether what you have is a monosaccharide or a disaccharide, then the test that you will be doing is a Barfis test. So here, monosaccharides react very fast, so within 2 to 3 minutes, whereas disaccharides react very slowly. So that is why when doing Barfis test, you must really make sure to take note of the time. Because if you will not be taking note of the time and a positive result occurs, then you might be observing it wrong, or you might be collecting a wrong data. Because, for example, you forgot to take note of the time and the red precipitation has occurred, and then the red precipitation has really taken place or was really produced within 2 to 3 minutes, but you were only able to observe it about or after 5 minutes, then you might conclude that what is present is a disaccharide instead of a monosaccharide. So Barfis test is done timed. So in Barfis test, the positive reaction indicates the presence of a reducing monosaccharide if the precipitation has taken place within 2 to 3 minutes, because on prolonged heating, disaccharides can also give a positive result. Hence, the solution should be boiled only for 3 minutes. And the positive result is the formation of a brick red or a dark red precipitate. For our next test, we have Seluwanovs test. So ketohexoses on treatment with hydrochloric acid forms a 5-hydroxyl methylfurfural, which on condensation with resorcinol gives a cherry-colored complex or a cherry red-colored complex. Actually, Seluwanovs test is a test which gives a positive result if ketohexoses are present. So specifically, if a fructose is present. Now, for our qualitative tests, Seluwanovs test is actually a confirmatory test for fructose, and it helps us to distinguish between fructose and glucose. Now, overheating of the solution should be avoided. Why? Because upon continuous boiling, aldoses can be converted to ketoses and will give a positive reaction with the reagent. So for Seluwanovs test, these are the two results. So here, this one indicates no reaction. So that means that ketohexose is not present in the solution. So therefore, that is not a fructose. Now, if this color is produced or a cherry red or a dark red color, indicates a positive reaction. For our next test, we have the benedicts test. Now, benedicts test is also a test for reducing sugar. Now, if you can still remember, Barfis test is also a test for reducing sugar. Now, what is the difference between the two? Barfis test is a test that can help us identify a monosaccharide from a disaccharide. Now, benedicts test will only help us to determine whether our unknown carbohydrate or unknown solution is a reducing sugar or a non-reducing sugar. So Benedict's test is usually done after Barfis test has been done. So this will just be a confirmatory test, for example, for sucrose. It will help us to identify if what we have is a sucrose or a reducing sugar or a non-reducing sugar. Carbohydrates with free aldehyde or ketone groups have the ability to reduce solutions of various metallic ions. So this one is also a semi-quantitative test because the color of the precipitate or the color of the solution will give us a rough estimation of the amount of the reducing sugar present in the sample. So here are the positive results for the Benedict's test. So for example, after the reaction, a green color, because originally the Benedict solution is actually blue. Now, after the test, for example, the solution turned into green, then that means that there is a presence of a reducing sugar. Now, and then, for example, green precipitate, yellow precipitate, orange precipitate, and the brick red precipitate. Now, if what you will be observing is the formation of a brick red precipitate, then that means that, okay, compared to a solution that has turned into a green color, then the solution where you observe the formation of a brick red precipitate means that the concentration or the amount of reducing sugar present is greater compared with that of a solution that has changed into a green color. So these are the results or the example for a Benedict's test, for example, for a negative reaction or no reaction, which means that no reducing sugar is present. Now, for a positive reaction, this is one. For example, the solution has turned into an orangey or a rusty colored solution, and the formation of a brick red precipitate will mean a positive reaction or a positive result. Next, or the last test, is the Oceazone test or the Oceazone formation. Now, this Oceazone test is actually the confirmatory test. So, for example, if you're trying to figure out what your unknown solution is, you can proceed right away with doing an Oceazone test or an Oceazone formation. A solution of reducing sugar, when heated with phenylhydrazine, characteristic yellow crystalline compounds called Oceazone are formed. Now, among the four different types of, or among the four samples of sugars or carbohydrates that we have used, so we have glucose, fructose, lactose, and sucrose. Only sucrose is the non-reducing sugar. So if, for example, if our unknown solution is a sucrose, then we will not expect a formation of an oxazone or a yellow crystal because oxazone test will only be able to form, or in oxazone test, a yellow crystal will only be formed if our sugar sample or carbohydrate sample is a reducing sugar. Now, how will we be able to know what reducing sugar is present if, for example, a yellow crystal was formed? Now, these crystals have actually definite crystalline structure, precipitation time, and melting point for different reducing sugars. So when a crystal has formed, we can look at it under the microscope, and the specific shape that will be formed will help us to determine what specific reducing sugar is present. These are the examples of the crystals that will be formed. So the first one here, we have a needle-shaped crystal or Oceazone crystal under the microscope. Then this needle-shaped crystal is for glucose and fructose. Now here we have a sunflower shaped. Now this is for the maltose carbohydrate. This one, the galactosazone crystals, is viewed under the microscope. And we have a powder puff or hedgehog or cotton ball shaped crystals for lactose. So these are the different shapes of the Oceazone crystals observed under the microscope for the different reducing sugars. Okay, so here we have a summary of the reactions of glucose. So for Molish test, the observation is purple ring at the junction of two liquids. Inference, glucose is a carbohydrate. Now for Benedict's test, upon observation, a brick red precipitate is actually formed, but it will depend on the amount of sugar. Glucose is a reducing carbohydrate. For Barfis test, scanty red precipitate at the bottom of the test tube, or a brick red precipitate or a dark red precipitate, which means that glucose is a reducing monosaccharide. Next test is Seluwanovs test. It will not give a positive reaction for Seluwanovs test, so therefore there will be no cherry red color that will be observed. That means that glucose is not a ketohexose. Now for Oceazone test, it will give, or what will be observed is a needle-shaped crystal, which is a confirmatory test for glucose. Now this is the summary for the observations and inferences for the reactions of fructose. And here is a summary for the reactions of lactose. Now, if you will notice, there is no Seluwanovs test for lactose. And if we will be doing a Seluwanovs test for lactose, what we will be expecting is that there will be no reaction, or there will be no formation of a cherry red color, because lactose is not a ketohexose and it is a disaccharide. And lastly, we have the summary for the reactions of sucrose. Now, if you will notice in here, for the reactions of sucrose, it will give a positive reaction for the Seluwanovs test. Why? Because sucrose contains, or sucrose is a disaccharide, which is formed from the condensation of two monosaccharides, which are glucose and fructose. And fructose is a ketohexose, so then it will give a positive reaction for the reaction of sucrose. Okay, so that's it for our tests for carbohydrates.